We have two quick things that we're gonna tick off from the end of the schedule today. First, I want to introduce Jonathan Rothberg. He is he's from Ion Torrent and the founder of 454, and he's going to I think give away a machine. Give away a sequencer.
Nothing makes you more popular than giving things away free. Thank everybody. I do want to thank obviously George Church and the organizers. I'm going to go pretty quick on the Ion Torrent story. And basically, my story starts personally. My son was having difficulty breathing, and I envisioned 454 sequencing. But when I think about that, and while I'm very, very proud of that, I really started something that was the mini computer age. And it allowed about 200 labs to do some really amazing things. And I've always been inspired by Watson and Moore, and my goal in Ion Torrent was to bring their work together in the personal computer equivalent of a sequencing machine. And in a personal computer, the machine is the chip at the core. And we were talking about large industries. What we decided to do was leverage the $1 trillion supply chain that has been created by the semiconductor industry and leverage the 45 years of compounded Moore's law. And what we created was a sequencing machine on a chip. And it has all the advantages that allow low-cost electronics, specifically the ability to scale. And it's really the simplest sequencing in the world. So the entire machine is here. There's no lasers. There's no optics. And the second trick is Watson-Crick base pairing, which gave us obviously the basis of heredity, but also the basis of the Ion Torrent sequencing. And what we do in millions of simultaneous reactions is synthesize a complementary strand of DNA. And we read off the sequence by detecting the hydrogen ion. So natural bases, no imaging, everything natural. And I like to say it's Watson meets Moore. Moore gave us this incredible semiconductor industry that allows us in over 400 factories around the world to produce this wafer that has 300 sequencing machines on it. Allows us to go from these wafers to any quantity of packaged chips ready to put into a machine and run without any human intervention, and at a cost structure that's unmet by any other technology. So we basically flow over millions of simultaneous wells, natural bases. We watch the extension on the bottom of these sensors. All the electronics are on the chip, and you can sequence genomes. We've sequenced the first post-light genome since Sanger, which was the first genome. Every genome ever done either had X-rays and or light. We sequenced the adenovirus as a proof of principle to 6 nines of accuracy, and we wanted to show its scales, so we sequenced a bacterial genome. What we're doing, though, is doing something that the PC industry did. We're going from being a vertically integrated company to a horizontally integrated company. And the reason we offered a contest is what we're looking for is killer applications. And what we've done is plugged into an ecosystem that has bioinformatics and upstream molecular biology, and we're part of that ecosystem, allowing any researcher anywhere in the world to immediately sequence DNA cheaply and inexpensively. In fact, the machine that's up here is going to retail for less than $50,000, and the experiments are less than $500. So now there's no reason not to own a machine. There's no reason not to do an experiment. In honor of Watson and Moore, we're giving away 2 of these machines. We held a contest. We had a few hundred applications. I want to thank again George Church and Ron Davis for reading all of these applications, and we chose 2 of them that exemplified the spirit of Watson and Moore, and I'm going to introduce you to those winners now. The James Watson Healthcare Grant goes to John Iafrate and Long Lee of Massachusetts General Hospital, and this is just fantastic. These are people that look at hundreds of tumors every week in a clinical setting to inform physician decisions, and we felt there's no better place to have an impact of the ion machine than right there, so we can look at over 500 oncogenes for those patients, and they'll say a few words today. The second is in the spirit of Gordon Moore, and Gordon not only gave us Moore's Law and signed a wafer for my son. This is the Jim Watson signature. But Gordon is also a big environmentalist, and in honor of Gordon, we're giving away the second award to Mitch Sogan, and Julie Huber will be accepting that award. And what's important here is somebody said, well, why are you doing water? And when we looked through these hundreds of applications, there was no application that we could find that had a greater societal impact. Every day, 14,000 people die because of contaminated water. So we felt that putting these machines in laboratories to have an impact on cancer and fresh water would exemplify what we're doing at ION. We're trying to take all of this information from the human genome, and we want to democratize sequencing in the same way that the microprocessor democratized computing. The ion semiconductor chip, we hope, will democratize sequencing through not only their work, but the hundreds and ultimately thousands of researchers that we will supply our machines to. So I want to call up first Dr. Long Lee of Massachusetts General Hospital, and he will be receiving the Watson machine. Again, I want to thank Jim Watson for signing the ion sequencing machine. Thank you.
Well, good evening. My name is Long Lee, and I work with Dr. John Iafredi in the Diagnostic Molecular Pathology Laboratory over at Massachusetts General Hospital, so really just a stone's throw away across the river here. Unfortunately, John couldn't stay for the entire evening, so in his absence and on behalf of our lab, I am truly delighted and honored to receive this grant award for one of Iron Torrent's personal genome machine sequencer. So if you don't mind, I'll just give you a little story about our background and how we arrived here today. So our lab is truly a clinical molecular diagnostics lab. We do some research, but most of our work is in the clinical arena. And we use a variety of techniques for cancer diagnostics. So since the start of our lab in 2005, we've realized the importance of cancer genotyping for diagnosing the disease itself, for providing any prognostic information, and also for determining mutations that might be used to stratify patients into molecular targeted therapy. So last year we launched a cancer genotyping platform that you may have heard about, based on a technology called Snapshot from Applied Biosystems. So using this system, we are able to generate an assay that can detect 100 point mutations. So initially we started in the lung group, we moved to the GI group, and now we're opening it up to all brain cancers at MGH. Essentially our goal is to allow this test to be available to all cancer patients that come through MGH. So in the modern era of genomics, and in the context of today's meeting, genome sequencing, you might think 100 point mutations is a measly small number. But you might be surprised to also hear that we're only one of very few labs in the nation that's actually doing such a multiplex test for cancer diagnostics. But we realize the limitations of our current technology in that we can only look at point mutations. We can't get the full breadth of cancer mutations that exist out there. So we're talking about copy number variants, methylation patterns, small indels, and mutations that are scattered throughout exons in various cancer genes. You can't capture that with a point mutation assay. So to address this issue, about 2 years ago, I did a molecular genetic pathology fellowship and was sitting in John's office, and we were tinkering with the idea of, what if we can use next generation sequencing for cancer molecular diagnostics? Fast forward 2 years later, about 2 months ago, I'm still in John's office. We're still tinkering with the idea of using next generation sequencing for cancer diagnostics. So what has changed in these 2 years? What has changed is that there's been a number of companies that have popped up here and there in terms of next generation sequencing. But in our opinion, what hasn't changed is that we don't think there's one platform out there that can really truly be applied at a clinical level. So about 2 months ago, I lurked around various blog sites, Genetic Future, Seek Answers, Genome Web, and also followed the ABGT meeting through Twitter. and caught attention of this entity called Ion Torrent. So what is Ion Torrent? Is it just another website for downloading stuff? Or is it truly a next-generation sequencing platform? So obviously, a little bit of research, we found out that a 1 to 2 hour runtime, a footprint that's really small, we can actually fit in our lab. It's low cost, so easy for scalability and high volume testing. Simple data structure directly from the chip, no image data, so a small lab like us wouldn't have to worry about the storage issues. 100 base pair reads and a partnership with CLC Bio. So all these were basically merits and advantages that we think can truly be applied at the clinical level. So what we propose to the company is this. We're going to cast a much wider net in terms of cancer mutation detection, about 500 to 1,000 exons. Not a lot, but these are the targets that we think are clinically relevant. These are the targets that are actionable cancer genes. And by actionable, what we mean is that either this mutation will clinch the diagnosis, it will give prognostic information, or more importantly, it will stratify patients so that we can give them truly personalized cancer molecular therapy. And most importantly, we want to validate this test in a CLIA certified lab. We want to make this accessible to all 100 or so tumor patients that come through MGH per week. And we want this to be a test that's not, in terms of the results, not sitting in a research lab for 2 months. We want this test to have a turnaround time of 2 weeks because we want the data to be in the hands of clinicians so that they can make important treatment decisions on the fly. So for us, it's not about sequencing the next full human genome. It's actually bringing this new exciting technology, which is called next-generation sequencing, to the individuals that we think need it most. Those are our beloved ones, our family members, our friends who are diagnosed with cancer, and that we truly believe that with such a broad-based clinical genotyping diagnostic assay that we can help them not next year, not tomorrow, but if we had an assay in the clinic today, we can truly help them in terms of their disease. So if a small lab like ours can get access to this technology, you can just imagine how quickly this can be implemented in terms of the clinical level. So last Thursday, John and I were sitting together again in his office after talking to Ion Torrent. So obviously we weren't tinkering with the idea anymore of next-generation sequencing for cancer molecular diagnostics, but instead we were actually planning experiments to actually achieve this and achieve this today. So we would like to thank Ion Torrent for their confidence in our work. And we look forward to collaborating with the company as well as any other interested parties who would like to turn our idea into reality. Thank you.
While we're doing these photographs, let's get up.
Yeah, I want to invite Julie Huber to come up. Mitch Sogan was able to have dinner with us last night, but he had a previous engagement, was unable to make it. And he did say Julie's a better speaker.
Oh, that was nice of him.
Thank you, Julie.
So I really think I'm the last woman standing between you and the bar. So I'll keep it as brief as possible.
Not true one.
What?
There's more. So.
Oh, okay. I'll keep it really brief. But on behalf of all of us at the Marine Biological Laboratory at Woods Hole, we want to say thank you. It's a pretty exciting opportunity. We want to drive the home machine home tonight, if that's possible. We're really excited to integrate this technology into our sequencing facility, into our microbial ecology laboratories, into our experimental schemes, and we have had a very open access platform with sharing next generation sequencing technology, particularly with microbial ecologists that don't always have access in their own labs. About 5 years ago at the MBL, we pioneered efforts to use next generation sequencing technology to start profiling microbial communities. You heard a lot about that from Rob Knight earlier today. Initially, we were focused on the marine environment because that's where our expertise lies, and we were part of the Sloan Foundation's Census of Marine Microbial Life. And with that effort, we have profiled over 1,200 different habitats in the world's oceans. So we have community composition data, we have diversity data, we have relative abundance data. We've discovered thousands of new microbes. We conservatively estimate there are tens of millions of different microbes in the world's oceans. And with this sequencing strategy, we sort of rediscovered something called the rare biosphere, and with the ability to routinely detect and monitor low abundance organisms in our environment, we very quickly realized there were many more applications of this technology beyond the marine environment. So since then, we've done numerous complex sequencing profiles from underwater volcanoes, which is actually what I mainly study, to the human gut, and more recently to sewage outfall. So we're now interested in applying this new technology to developing both cost-effective and rapid methods to profile microbial communities that may pose a threat to human health in our water distribution and our recreational water environments. So the ability to carry out what we sort of call pathogen surveillance, which we've heard quite a bit about today with sequencing, is that you can get much more complex signatures of organisms and groups of organisms and relationships to their environment. And that's going to be the main thrust of our application of this instrument. So traditionally, for water quality monitoring, we use a very old-fashioned, tried-and-true method of going out, taking a water sample, and trying to grow an indicator organism, most commonly E. coli, in our laboratory. They're very easy to grow, but obviously when you're just looking at one or two indicators, you're very limited, and you're also not necessarily tracking a true pathogen in the environment. This method, it also takes at least 18 to 24 hours. So there are often restrictions on drinking water supply or often beach closures, for example, that are not necessary or they're simply too late. So with this platform, it's rapid. We can do 500,000 reads, my understanding, in an hour for about $500. And that begins to bring the cost per sample very close to the cost of what we've been doing with these traditional cultivation methods. And so we're beginning to think that this could be the type of technology that is routinely used for things like water quality monitoring. Someday, this is one of my pie-in-the-sky dreams, one could even imagine having this in a field deployable package where you never even had to go back to the laboratory. I often go out on research ships in the middle of the ocean for long periods of time, and I would consider bringing something like this with me. But the idea to be able to instantly get that answer out in the field, right now we're going to be able to do it now in a few hours. And so that's sort of our vision of where we hope to see this go. And the idea is to be able to identify indicator pathogens, suites of organisms in the environment, novel discovery, so that we can track and monitor our water supply much better than we can. So thank you for this opportunity, and we can't wait to get it.
Thank you. I want to wrap up by thanking again George Church and Ron Davis for judging these applications with us, and congratulations to the winners. And maybe if we come back in a year or two, George will show one of these chips in a handheld unit in front of everybody. Thanks.
Thanks.